So what do the most favorable conditions tell us? Well, as a recap, in video 1.2, we found out that mathematical models aren't useful in biology. And I bring this up because I made that statement, and I want to acknowledge it and say it's still valid. And if you want to understand it, please go to video 1.2 to review that content. The scientific mathematical model that we're going to talk about is not just including biology. It includes 3 disciplines: chemistry, information theory, and biology. The information theory builds the mathematical model. When we say information, we mean a measurement about the amount of information that is contained. You can actually measure information. That's the math part of this. Chemistry is used to describe the processes, and biology provides the empirical parameter values that are used to make the calculation. So we're going to build a scientific mathematical model at the intersection of these 3 disciplines. What are we going to model? Well, the human genome. The human genome is the complete heritable information of a human being. There's quite a lot there. You have 23 chromosomes, about 3 billion base pairs. It's 6 feet long, 20 to 30,000 genes that produce a million protein variants. And what we need to show is that undirected macroevolution is capable of producing the human genome by chance. We're going to make the best case approach. Our mathematical model is just going to be out the information in the human genome. We're going to focus on the genetic code and ignore the other codes. This reduces our work. We're going to focus on just the information used, and you'll see why that's important in a minute. We're going to use the most favorable conditions and assumptions. We'll use real-world measurements where possible. And our analysis, we want it to be as simple as picking a green ball from a bag of colored balls. We don't want our analysis to be so complicated that we're unsure about its conclusions. We want surety about the conclusion. So our approach is to assume the genetic code exists. We're going to ignore an explanation for the creation of the genetic code. We're going to use a simple information model of the DNA, and by simple I mean we're going to assume one gene maps to one protein, the central dogma. We'll calculate the information content of DNA, focusing on the used information. Then we'll calculate the probability of the used information arising by chance, and then compare that calculated probability with other probabilities using the nonsense detector. To kick this off, we're going to have to discuss what a general communication model is. I will make this as easy as possible for you, so please come along this journey. We have a data source, an information source that sends the information to an encoder and a transmitter. The transmitter sends it out over a channel. There's noise that's added to the channel. The transmitted noisy data is received and decoded, and then the destination gets the information it needs. So information comes from the source, information is received at the destination, and noise is added to the information in the channel. Now this sounds really abstract, so let's make it more concrete. Let's look at a communication model of a radio. On the left side, we have a microphone, and we want to transmit CD quality audio. That microphone signal goes to an amplifier, an A to D converter. It will convert it to a digital bit stream. That is sent out over radio waves. Noise is added to that signal. A tuner receives the bit stream and converts it back to an analog signal, which is then sent to the speaker. In this case, for CD quality audio, the input data is 242 kilobytes per second. There's some error correcting data in that signal. of 66 kilobytes per second. And so the actual received output data is 176 kilobytes per second because the error correcting data doesn't provide any value to the output. Now, I want to point out that these input data, error correcting, and output data rates are the maximum capacity of the channel. If nobody's speaking in the microphone, then the input data will just be 0. It will still send out the error correcting 66 kilobytes per second, but the output data will be 0 again. And so the actual usage can be a lower byte rate. Let's talk about a television. We have a camera with 1080p quality video. Its signal is sent to an amplifier and video codec. It's sent out over radio waves. Noise is added to the signal. A receiver receives the radio waves. A video decoder converts it to a video screen signal. In this case, the input data is 10 megabits per second. There's a little error correcting overhead or signaling overhead of 0.3 megabits per second. So the output data that's usable is 9.7 megabits per second. And the screen will actually have an uncompressed rate of 746 megabits per second. And the reason it does this, or can have such a high rate, is that it actually stores the data that's sent into it. And so what is sent here in terms of the input data would be the changes that are made to the output screen. So again, this is the maximum capacity of the channel. If it's midnight and there are no lights, the camera will have a black background. There will be no input data. There'll still be the error correcting overhead and no resulting output data. And you'll see a black screen. Okay, so we've done this. Why did we do this? Well, because we're going to look at a communication model of protein transcription and gene expression. This is based on the research of a gentleman named Hubert Yockey. We see this paper is from Computers and Chemistry in 2000. In that paper, he shows this communication model. On the left, it has the message in DNA code. You have transcription. The mRNA message is sent through the communication channel. Noise is added to it. It's decoded by translation, and a protein is generated. So the source is DNA. The output is the protein. Well, this is what that actually looks like in real life. You have the DNA with RNA polymerase, messenger RNA is sent. You have key enzymes and factors working with the ribosome, transfer RNA, all of that to generate a protein. That's what a communication channel looks like when we're talking about gene expression. You have DNA in and protein out. This is the sort of real world way of describing this. We can try to estimate the probability of all of these physical processes working by chance, including all of these things you see here on the screen, but that's really too complex to estimate. We can't do that from a practical standpoint. So we're going to use a simple method, which I hinted at before. We're going to get an optimistic probability by ignoring all of these physical things. We're going to estimate the probability based on the information used. Now that probability based on the information used is going to be rather optimistic, but we'll see that it doesn't really matter in the sense of the optimistic probability is going to be very, very small. So our communication model of gene expression is we have the DNA code, we have transcription converting DNA to mRNA, we have some mutations that occur due to genetic noise, then translation, and out pops a protein. So given our communication model, how much information is involved? We have three values we'd like to understand. What is the maximum data that can be sent through the communication channel and how much of that is the output data, typically how much information is used, and what's the minimum amount of information that's used. So how do we calculate the maximum usage of that information channel? We turn to a paper by Dr. Yockey. Notice this paper is from 1974. He came up with this idea over 50 years ago. This is the initial paper that described his findings. And in there, he talks about three values. 5.833 bits per codon. That's the maximum information that's input. He talks about 1.680 bits per codon, which is the error correcting information, and 4.153 bits per residue, which is the output information. So we plug that in. We find the input data is 5.83 bits, error correcting is 1.68 bits, output data is 4.15 bits. That is the maximum amount of information that can be pumped through this communication channel. What we're interested in is what is the typical and the minimum usage of that information. What we need to understand is how much information is needed for the protein. We know there's redundant data for error correcting, so we can subtract that. But we also want to note that different proteins can have the same function. What we mean by that is, even if there is noise in the channel, and that noise is so high that the protein that was intended is subtly different, that resulting protein could still have the same function. And so there's some additional built-in error correcting if you allow for proteins having the same function. We need to remove the amount of data that contributes in both cases. We again turn to Dr. Yockey to find out the minimum and typical channel usage. This is from a paper of 1977. He looked at the cytochrome C protein, and what he found is over a huge sampling, he found that it's 3.701 bits per residue that typically is used. Then he did some analysis and calculations, and he reduced that to 2.953 bits per residue. So based on this data, we can show that we can select the output data as 3.70 bits typically, but the minimum is 2.95 bits. For our purposes, we want to use the most optimistic value, so we will use 2.95 bits in our calculations. We're now ready to calculate the maximum amount of information in the human genome. Now, the maximum amount of information is the information that's used, but also additional, call it junk information. And what we want to focus on is the used information, but this is the first step. We're going to ignore duplicate chromosomes. We're going to use the most recent research regarding the number of base pairs in the human genome, which is about 3 billion base pairs. There's one protein residue per codon. If we take out the information used for error correcting, we use the minimum information per codon, which is 2.95 bits per codon. We do a little bit of math, simple math, and we find that the human genome has 3 times 10 to the 9 bits of information. But again, we have the used information, and then we have this junk information, and we need to figure out how much of that 3 times 10 to the 9 bits of information is actually used. Now, one of the things we've talked about is that there's some useless information or duplicated data in the DNA. So we need to account for that. So how much information is used? We turn to the ENCODE project, and we turn to this paper called An Integrated Encyclopedia of DNA Elements in the Human Genome, published in Nature. And what it says is the vast majority, 80.4% of the human genome, participates in at least one biochemical RNA and or chromatin-associated event in at least one cell type. We can calculate how much information is used. We know that 80% of the human genome is used. If the human genome has 3 times 10 to the 9 total bits of information, then doing a little bit of calculation, the human genome has 2.4 times 10 to the 9 bits of used information. The next step is to take this amount of information we've calculated and convert it to probability. Again, we're just using a simplified information model and ignoring the more complex physical processes. We're going to focus on just information. We're going to use our simple information model to calculate a simple probability model. We're going to take those 2.4 random bits and we're going to calculate the probability of them matching against a pre-specified sequence. This needs to be calculated by chance, and so we're going to randomly select a 0, 1, do that 2.4 billion times. If we were to do that, the calculated probability is approximately 10 to the negative 722 million.